Today I'll be sharing insights from my research which explores how cutting-edge computational tools are revolutionizing the drug discovery process. Specifically, I focus on bridging the gap between AI-based and physics-based methods. This work was conducted at the University of Toronto under the Structural Genomics Consortium and supervised by Professor Shapira. Developing a new drug nowadays is truly a monumental task. On average, it takes over a decade and costs between $1 to $2 billion to bring a small molecule drug from concept to market. This is a very lengthy and expensive process which can pose significant challenges in addressing urgent medical needs. The traditional drug discovery pipeline involves multiple stages. First, identifying a target, then discovering molecules that bind to the target, so hit identification, a hit to lead optimization, and a lead optimization, which finally leads to preclinical testing and clinical trials on real humans. Every single phase is very time-consuming and resource-intensive, contributing to the overall complexity and cost of drug development. In recent years, computer-aided drug discovery has really emerged as a transformative approach, enabling researchers to screen vast libraries of compound virtually, thus accelerating the identification of potential drug candidates. Applications are introduced at many steps of this process. Computers can aid in target validation through simulations or hit identification through computational screening and docking tools. So a really groundbreaking advancement in the field is the AlphaFold, and this is an AI system developed by DeepMind. AlphaFold predicts the three-dimensional structure of proteins with really remarkable accuracy solely based on their amino acid sequences. So this capability has been a game changer in the entire field of structural biology. AlphaFold has now predicted structures for over 200 million proteins, covering nearly all cataloged proteins known to science. This is an extremely vast repository and helps accelerate research across various domains, from understanding disease mechanisms, for example, to developing new therapeutics. Now let's take some time to dive deeper into AlphaFold 3, which has recently been made available to the public and is arguably one of the most exciting tools in modern computational biology. AlphaFold 3 builds on the groundbreaking success of AlphaFold 2, which was able to predict protein structures from just amino acid sequences with near experimental accuracy. While AlphaFold 2 focused on single-chain protein structures, AlphaFold 3 is designed to model more complex biological assemblies, including protein-protein, protein-DNA, and protein-ligand interactions. What makes AlphaFold 3 so powerful is, at its core, AlphaFold 3 uses deep learning, specifically this transformer-based architecture, and transformers are the same class of models that power tools like ChatGPT. They're very good at learning long-range relationships in sequential data, and in the case of AlphaFold, the sequence isn't natural language, but it is a sequence of amino acids in a protein. Looking closely at the general architecture of AlphaFold 3, we have a multiple sequence alignment module, which is going to give the model insights into how residues co-evolve and help infer their spatial proximity. AlphaFold can also use structural templates when available, referencing known protein folds to improve its overall accuracy. The pairformer handles information flow between amino acid residues using attention mechanisms. It processes pairwise residue information, sequence level features, and ligand and covalent bond information. We can say that the pairformer is at the heart of AlphaFold 3's reasoning engine, and it's going to learn these relationships between residues and pairs. After learning these relationships, the diffusion module predicts the 3D coordinates of every atom in the protein through diffusion-based generation, a technique inspired by diffusion models in image generation. This allows the model to iteratively refine 3D atom positions in a generative fashion. The diffusion module will bring flexibility and robustness to 3D structure prediction, which is especially useful when modeling complexes, small molecules, and more flexible interfaces. Other than the ligand and complex prediction being a new feature in AlphaFold 3, we now also have confidence scores, so the model outputs not only structures but also confidence metrics, which will tell the researchers how reliable each part of the structure is. AlphaFold 3 essentially collapses years of experimental structural biology into minutes. But, and this is where our study fits in, these predictions are static. They represent perhaps a single low energy pose. It is important to keep in mind that protein and ligands are still dynamic and flexible systems. To address this, we thought of using molecular dynamics simulations to come into play. And by simulating the physical movement of atoms and molecules over time, MD provides insight into the dynamic behavior of protein-ligand complexes, offering a much more comprehensive understanding of their interactions. In an MD calculations, one has to assign initial values for each of the internal and external coordinates of each of the N molecules, and an initial value for the kinetic energy or momentum of each coordinate, after which a time propagation algorithm will generate values for the coordinates and momenta at later times, with each coordinate having an initial velocity and values specified. One uses Newton's equation written for a time step of duration delta t to propagate forward in time according to, for example, this first order propagation formula. Right here, Gaussian accelerated molecular dynamics, or GAMD, is designed to enhance the sampling efficiency of classic MD simulations by adding a biasing potential. Energy barriers that would typically slow down this exploration of the system's conformational space are overcome, and so this ultimately allows for the system to explore different states much more quickly. In our study, we integrated AlphaFold 3 predictions with Gaussian accelerated MD simulations to refine the modeling of protein-ligand interactions. We applied this combined approach on 3 distinct protein-ligand complexes. I've included the relevant structures in the slides. So right here on the left is WDR55, and it's involved in ribosomal biogenesis. We investigated its binding affinity with 2 different ligands. On the right is HAT1, which is an enzyme that acetylates histone lysine residues. And ultimately, we aim to assess the strength and limitations of MD simulations and refining the AlphaFold 3 predicted protein-ligand structures. Now on to the general workflow. So the protein-ligand complexes were selected for investigation and experimental structures were generated by X-ray crystallography. And to model the selected complexes, AlphaFold 3 was installed on an in-house cluster. For each protein-ligand complexes, amino acid sequences, and the corresponding co-crystallized ligand smiles were used as the inputs for AlphaFold 3. Default settings were used except for the number of seeds, which was set to 5. For each input, AlphaFold 3 writes 5 samples for each seed, and at the end of the prediction ranks all 25 samples and scores them with a ranking score. The top ranking molecule is presented as the most confident prediction. Gaussian accelerated MD simulations were performed using the newest version of NAMD. Protein-ligand complexes were prepared using the CHARM GUI enhanced sampling module. Each system was solvated in a rectangular box using the TIP3P water model with a 10 Armstrong edge distance from the solute. The system was neutralized and ionized with sodium and chloride ions placed via Monte Carlo. Ligand parameters were derived from the CHARM general force fields, and protein and solvent parameters were applied using the CHARM 36 force field. The simulation protocol in general involved first energy minimization, then equilibrium under the canonical ensemble, and finally production under the isothermal isobaric ensemble. The simulations were conducted with periodic boundary conditions with a 2 femtosecond integration time step, and temperature was maintained at 300 Kelvin with pressure held at 1 atmosphere. Production MD was performed in triplicates for every single complex, with the total simulation time being 200 nanoseconds for HAT1 and 300 nanoseconds for the WDR55 complexes. After the simulations, the 2 most statistically likely conformations from the MD simulations were selected as cluster 1 and cluster 2. So protein-ligand interaction were visualized and analyzed using PoseView to generate 2D interaction diagrams highlighting intermolecular bonds and binding interactions. The root mean square deviation, or RMSD, of ligand poses was calculated using PyMol to assess the structural deviations between predicted and experimentally determined structures. RMSD values were calculated based on the alignment of the ligand-heavy atoms between the different conformations. Here is the general formula for a root mean square deviation calculation. Now on to the experimental results. So experimental AF-3 predicted and MD refined clustered poses were overlaid here for visual comparison. In pink we have the experimental structure, in green the clustered structure from MD simulations, and in blue the AF-3 predicted structure. RMSD analysis from the MD simulations indicates that all three protein ligand complexes exhibit relatively stable protein backbones with RMSD less than 3 angstrom. This corresponds to the blue graph here in every single plot. So typically a structure is considered very well equilibrated when backbone RMSD fluctuations are less than 2 angstrom. The time evolution of RMSD in general suggests that the AlphaFold3 predicted HAT1 structure has the weakest kinetic stability, as indicated by the very large RMSD fluctuations over the simulation time. AlphaFold3 predicted WDR55 thiophene, on the other hand, likely exhibits the strongest kinetic stability, as reflected by the lower, less frequent RMSD fluctuations over time. We can say that the previous results are concurrent with the PyMol calculated RMSD values, which quantify structural differences between experimentally determined and simulated structures. All of the scores indicated here are calculated relative to the experimental structure. The HAT1 complex exhibits the largest RMSD difference between AlphaFold predicted structure, or this starting pose, and the MD refined clustered pose, while the W5TH ligand has the smallest RMSD difference across categories. This follows expectations, as we would expect the complex which exhibits the largest kinetic stability, so W5TH, to move and fluctuate less from its original state. Looking at the net AlphaFold3 RMSD values, we notice that the HAT1 complex has the highest deviation from experimental structure, while W5TH has the lowest RMSD. We could hypothesize that higher kinetic stability may be correlated in this case with more accurate AlphaFold3 predictions. Further energetic insights such as free energy calculations could be very useful for a more comprehensive view. Finally, now comparing across all categories, we see that refinement was truly only achieved for the WDR55 acid compound. For both HAT1 and W5TH, the MD simulations actually resulted in higher deviation from experimental structure. The RMSDs predicted were higher than the RMSDs derived after the simulation. Furthermore, it's very important to note that in structure-based drug design, an RMSD of less or equal than 2 Armstrong is indicative of successful docking. So according to this metric, neither AlphaFold3 predicted nor MD refined poses were actually successful across all three structures investigated. Let's move on now to the 2D interaction maps and intermolecular interactions. So we can see that relative to the HAT1 experimental results, the HAT1 AlphaFold3 predicted structure is relatively uncoordinated, missing a few hydrogen bonds and hydrophobic interactions, which are essential for structural stability. This could explain the low kinetic stability of the complex as observed in the RMSD plots. During the MD simulation, we see that this ligand rearranged to favor pi-pi interactions and hydrophobic interactions. From this initial investigation of HAT1, we We conclude that MD simulations might have limitations when the starting pose deviates too much from the experimental results as seen from the RMSD on the previous slide. And especially this could be difficult to realize in a reasonable time frame. Interestingly enough, in contrast, now looking at the WDR55 thiophene complex, we see that the AlphaFold 3 structure is slightly over-coordinated relative to the experimental structure, stabilized by an extra hydrogen bond interaction as well as some hydrophobic interactions. Remembering from both the Table 1 results as well as the RMSD plots, this ligand had the lowest deviation from the experimental structure, with the AlphaFold 3 predicted value being the closest to the experimental structure. We do see here that the ligands are relatively well aligned in terms of orientation, implying that the critical contacts may have been reproduced by AlphaFold 3. This could explain the high kinetic stability of the complex, and in this case, the very high stability of the predicted structure may have led to difficulties in refinement using MD simulations, once again in a reasonable time frame. The MD simulations in a way confirm the stability of the AlphaFold 3 pose rather than altering it. Finally, now looking at the WDR55 acid complex, which was the only one that achieved refinement. The RMSD relative to the experimental structure and kinetic stability was in the middle of both of the previously mentioned structures. The interactions predicted here are also, in a way, intermediate, with certain very good hydrophobic interactions and contacts established, but some key hydrogen interactions missing. In this case, molecular dynamics may have allowed the system to overcome a minor barrier, moving it towards the correct geometry. In fact, from the RMSD plots, it seems that towards the end of the simulation, the ligand stabilized into a different position, and we can speculate in a way that MD simulations can assist in refining structures when AlphaFold 3 deviations from experimental structures are modest. These conclusions are being drawn from a very, very small data set, and further investigation will need to be expanded on a larger range of complexes. To conclude, the study set out to explore whether molecular dynamic simulations could refine protein-ligand binding poses predicted by AlphaFold 3, specifically in the context of small molecules. While Gaussian accelerated MD did not successfully improve AlphaFold 3 predicted poses across the board, particularly under conventional structure-based drug design protocols, the results still offer really important insights. So just to recap, we analyzed 3 types of complexes: HAT1, which is a poorly predicted and unstable complex; the WDR55 thiophene complex, which is a relatively well predicted and stable complex; and finally, the WDR55 acid complex is an intermediate case. From this range, we observed that MD simulations show the greatest promise in refining structures that moderately deviate from experimental confirmations. In other words, where AlphaFold predictions are close but not quite accurate, MD can potentially bridge the gap. Looking ahead, there's a clear opportunity to expand this work, both by analyzing a broader set of complexes and by exploring additional MD methods. As we wrap up this work, I'd like to briefly highlight some really promising future directions of this work. One key limitation of molecular dynamic simulations is, in fact, time. They can require really extensive computational resources to capture these biologically relevant motions, and so to address this, these enhanced sampling methods like Gaussian accelerated MD, which we used, are a step forward, but even faster and more scalable approaches will be critical as we move forward. One exciting possibility right here is AI-assisted simulations. Deep learning models can guide or even predict conformational sampling, and these approaches would or could significantly reduce the need for long simulations while retaining this accuracy. Another direction here would be the integration of free energy calculations in the workflow. So although all of the previously mentioned steps do give us qualitative insights, free energy calculations could really provide a quantitative binding affinity prediction and help us rank directly these drug candidates. Finally, there could be an interesting correlation between prediction accuracy and simulation time, and in future studies we could systematically evaluate how long simulations need to run before we see convergence in pose refinement, which would help optimize both performance and efficiency. Together, all of these future steps could really bring us closer to truly integrating AI and physics-based methods for drug discovery, and not just for refinement, but for fast, accurate predictions of real-world binding events. Before I conclude, I'd like to take a moment to express my sincere gratitude to everyone who made this research possible. First, I'd like to thank the University of Toronto for providing the academic environment and resources that supported my growth throughout this project. I'm also very deeply grateful for Professor Shapira, whose guidance and mentorship were invaluable at every step of the process. A very special thank you to Dr. Aziza Boussele, I worked very closely with. Thank you for your patience, insight, and mentorship, which truly made this an incredibly rewarding learning experience. As this was a collaborative project, I would also like to thank every other member that contributed to the results of this study. Finally, I'd like to acknowledge the Structural Genomics Consortium for providing the collaborative platform and tools that made this research possible. Thank you.